Welcome to this session of Shadow the Scientist. My name is Jamaica Marshall, and I am pleased to serve as the Shadow the Scientist Programs Manager. Shadow the Scientist is an initiative that's brought to you by a team at the University of California at Santa Cruz. Today's session is Hawaii's Abyssal Plains, Minerals, Ecosystems, and Discovery aboard the research vessel Kilo Moana. Now, our science hosts are Dr. Kira Mizell, and then we also have Caitlin, Peter, and Marissa. We are live with them on board the vessel Moana, and let's go over to Kira to get us started. Kira, thank you for having us.
Yeah, of course. Hi, everybody. Thank you so much for joining us. You can see me here in my muddy overalls. Yeah, we were out here exploring the abyssal plains, which are some of the deepest parts of the world's oceans, about 200 nautical miles south of the big island of Hawaii. So we got on our ship and we transited down here about 40 hours. So it took multiple days to get this far. So everywhere we look around us is just sky and sea. We've only seen one other ship the whole time we've been out here, a few birds, and we are sampling parts of the deep ocean that have never been sampled before. So we're mapping and looking at sediments and animals and looking at the water column, all to learn a little bit more about the deep sea.
Wonderful. Before we proceed, I will say that we welcome your questions and comments in the chat at any time. Arjun and students there with Arjun and Christian as well. You can put your questions in the chat anytime, and we're going to have those answered around the last 10 minutes of the session so that we can spend as much time with Marissa and Caitlin and Kira as possible. So, Kira, you have an amazing staff. I'm sure I know that this is not all of your colleagues, but I had the pleasure of speaking with Marissa and Caitlin just before we got started, and they got me so excited about what we're going to see today, Kira. Could you introduce them? And then I'd love to hear from Marissa and Caitlin themselves.
Yeah, absolutely. Yeah, just for extra information, we have 18 science crew on board. That includes 2 technicians and a comm specialist, but everybody else is a scientist working on individual science projects.
Hmm.
And here with me, I've got Marissa and Caitlin, and I'll let Caitlin go ahead and introduce yourself.
Yeah, hi everyone. So I work for US Geological Survey. Kira and I are on the same team. We study seafloor minerals and sediments. I have a background in marine biogeochemistry. So that's biology, geology, and chemistry all mixed together. And I've done a lot of work studying how mercury and metals cycle through seawater. But a few years ago, I joined USGS, and now I look at how metals cycle on the seafloor.
And my name is Marissa, and I'm a fourth-year graduate student at the University of California, Santa Cruz. So I'm also a biogeochemist, and I study mercury cycling in the ocean and now sediments. So the samples I collect on this cruise will be actually the last chapter of my PhD dissertation. Very exciting.
That is very exciting. That's very exciting, Marissa, Caitlin. Wonderful. Thank you so much. And it is certainly an honor to be a part of this last chapter for your work. I hope you give us a mention in there real quick, if you can. That would be amazing. So I know we only have a short time with you, so let's get started. You're on a research vessel and it looks like is this a lab that you're in? Tell us about what we're seeing behind you.
Yeah, we're in a lab right now. We're moving to our next station, and this is the next place that we're going to deploy our box core to collect some sediment. So if you see us swaying a little bit, it's because the vessel is moving and it's a little bit rocky today. So we're kind of in between sampling. There's nobody in here now, but once we pull up a sample, everybody gets really busy and we're taking mud to different labs and processing things in different ways. But right now we're transiting to the next station. So this room is a little bit empty. We have several different labs on board. We have some control rooms where we can watch the equipment that's going overboard and coming back on board.
And we also live on the ship. So there are staterooms where we sleep and this area called the mess where we eat. And I think we showed the video
Yeah.
that shows the tour.
We give
Oh,
you guys
yes,
a little
please.
virtual tour, and then we're gonna go for a walk.
Oh, all right. Wonderful, wonderful.
Can you see my screen?
Yes.
So I think we're gonna. Here it is.
This is the Kilo Moana, operated by University of Hawaii. And we're on the back deck now. Going up to the O1 deck. This is the bow of the vessel. This is a really cool double hull vessel. So there's actually two bow sections, and that design helps keep us a little more stable, which is really good for deep sea mapping. This is a giant winch that controls our equipment that we put over the side.
Fast.
This is where I spend a lot of time. This is a cold laboratory. So the deep sea is really cold and really dark. And when we bring up samples, it's very hot here. We're in the tropics. And so the environment at the surface is very different than the deep ocean. So we have this cold van where we bring our samples to start processing them so we can keep conditions as similar to the seafloor as possible. So you saw that red light. If we want it dark like the deep sea, we can turn the red light on to work.
Now we're going down to the gym. You can work out, although everybody works so hard, they barely have any time to work out. There's the kitchen staff that keeps us alive and where we all share our meals together. You get to be really good friends with the people you sail with. This is the staterooms. Here's the lounge where we can watch movies or have meetings or hang out together when we're not sampling. It's nice to have somewhere to relax. There's the engineer's room. They have full toolkits. I mean, if anything breaks out here at sea, we have to be able to fix stuff. So in addition to the science staff, there's a full crew of about 20 people taking care of the ship.
And
That's the lab we're in. Those are our cups that we've shrank. And you'll notice a lot of coolers and stuff on the floor. And that's because although the lab spaces are here, we bring all of our own equipment. So if we need a microscope, we bring it with us. If we need a special instrument, we bring it with us. So we spend about 10 plus hours setting up the labs to be exactly fit for purpose for what we need to do.
Kind of pack up our labs at home, put them in boxes, and rebuild it on the vessel.
Wow, that was incredible. So you said that you actually bring most of your equipment with you, which is different than what I've experienced with other expeditions. The science labs are already on the deck, they're already set up and stocked. So what is that like to have to bring everything with you?
It takes a lot of planning, and like Kira was saying about the engineers, if something breaks on the vessel, you have to be able to fix it. And so we bring everything we need, and then we have to think about how that equipment might malfunction and what types of tools we might need to fix it. Sometimes, Marissa's done a lot of work analytically at sea. So she brings an instrument and she's running samples, and those instruments aren't designed to be run 24/7. And so they often break, and you have to bring everything you need to repair and fix.
Wow. I mean, that really, it ensures that you are very confident and knowledgeable about all aspects of the work that you do, because you're the engineer, you're the technician, you're the repair person, everything. And you're doing all the data gathering, sediment gathering, and analysis. It's like you wear many, many hats on this kind of expedition.
Yeah,
yes.
absolutely.
We want
So
to go on the back deck.
I was just about to ask, now that we've had a virtual tour, I'm eager for Arjun's class and for Christian to see what it's like to be out on deck.
Yeah,
Let's go.
check it out.
If you need me to adjust the camera, let me know.
I will. I will. And Maya, thank you for that reminder in the chat that yes, we welcome your questions and comments anytime. We will definitely get to them in the last 10 minutes of the session.
Here's some of the scientists.
Hello, hi. Oh, wow.
Right. Yeah. So this is our box score. Can you guys hear me okay?
Yes.
Okay. This tool is called a box core. You can see on the side, it's kind of like a big metal box. And right now it's open. And this thing on the side is the scoop. And so we pick up the box by this wire and send it overboard. And the depth what's our next station depth?
About 5,500 meters, which is almost like 5 miles.
3 miles.
Wow.
3.5 miles.
3 miles. And so to get 3 miles down is probably going to take 2.5 hours. So we'll be hanging out on deck. We'll be watching the wire speed. We attach something to the wire where we can keep track of the depth. And then once this thing lands on the seafloor, it's going to trigger this big scoop to close. So it lands on the seafloor, the box sinks down, and then this scoop comes and closes, and we get a cube of seafloor of mud and water on top. Then we have to wait 2.5 more hours to bring this thing back on deck. We hook it with some taglines, tow it back on board, set it down, and then we have to unscrew this box, pull it out, and see what we got. We just did this a couple hours ago, and unfortunately it came up empty. That happens sometimes. So it took 4 or 5 hours to collect this box, and there was nothing in it. We had a teeny little bit of mud that we were able to scrape. Now we move to our next station and we try again. So we have some cool maps from this area that we can study and try and plan where we want to get the mud. Sometimes it doesn't work. Sometimes it comes up so full there's water spilling over the top. You never know what you're gonna get. Yeah, this way. There's the ocean. So we have a team on board that is counting all of the animals in the mud, including small little worms or gelatinous creatures. So when we're done taking samples of the mud, we have some left over, and we have giant shovels. So we shovel the mud into this table that has a sieve, wash it through, and then we pick through and make sure there aren't any rocks or animals. Very high tech. This is how we collect seawater samples. So this is a big rosette. We call it the CTD. That stands for conductivity, temperature, and density. And so underneath are a bunch of different sensors. And when this tool is being lowered into the ocean, we get a live feed of data from those sensors. And so we can see how things like temperature and salinity are changing with depth. And then these bottles are open at the top and the bottom. So as this is going down through the water's kind of flowing through this open bottle. We're studying the sensor data and we're seeing different features in the water that we think are interesting and that we want to sample. And then once we get all the way to the bottom, we see what the water column looks like. We bring it back up to the surface. And on a computer, we can click and say, bottle number 16, I want you to close here. And there's a little mechanism on the inside that's going to flip open and allow the top and the bottom of the bottle to snap shut. And then it grabs us a sample of water at a particular depth. I think we have 24 bottles on this rosette, which means we can collect 24 samples from 24 different depths in one cast. And we're still doing the deep water. So the box core we were saying is going down over 3.5 miles. This thing is also going to go down 3.5 miles. So we're going to collect water from the deep ocean that's cold and dark and really salty and really different than the surface warm water that we have. we can show you the control room where we watch the CTD feed.
Serious. You can hear.
Another
Wow.
Wow.
So this is the control room, and this is the computer where we can see the CTD. So, I don't know if you can see on this middle panel here, we have oxygen concentration up on the X and depth on the Y. So this CTD only went down to like 1,600 meters. So like less than a mile. And you can see that the oxygen was really high at the surface and then decreased with depth. So we're able to see this live as the instruments are going down through the water column.
And then behind Marissa, there's a radio. So we're kind of talking to the deck crew, the people who are driving the winch. Marissa will say, please go down 100 meters and tell them when to stop so we can collect data. So this is a fun room to hang out in. Oh, I'm shorter than you. We're gonna have to adjust.
The videos for the other labs, or?
So now we'll go back into the lab we were in before. Oh. No. The Karen. Oh,
It's okay.
it's okay.
Listen, we're in the team's lab that's studying the fauna, the animals that live in the sediment. So they were coming in to prep some bottles, start doing some labeling for our next box score. We're crashing in their space.
Well, please tell them that we said thank you for allowing you and us to use their space. That was an amazing tour. Thank you so much. And again, students, everyone in our virtual audience, go ahead and add those questions into the chat at any time. And we can go with the flow, Marissa, Kira, Caitlin. I see we have a question that just popped up. If you'd like to answer that now, we can, or I can hold it to the end. How do you want to proceed?
Yeah, that's fine. Yeah, so the question is, how many box scores have we pulled up? That's a great question. We are transiting to box score site 21. So we have deployed the box score 21 times. There's been a couple of times, like Caitlin mentioned, that it's come up empty. There's also been a couple of times that the box score mechanism failed, but we've deployed it 20, coming up on 21, or we have deployed it 21 times, and we'll recover the 21st box in about, I don't know, an hour. So we're getting really good at practicing the deployment, and it's a really exciting thing. I mean, the box score weighs about 2,200 pounds. So it's a giant metal box swinging all around, and for the most part, it's under control, but it's a pretty adrenaline rich activity. So it's like, if you like excitement mixed with your science, oceanography provides those opportunities to really get your blood pumping, not just from the excitement of seeing cool chemistry and sediments and animals, but also from wrangling giant equipment on the back deck of a moving ship.
So
There's a couple more.
yeah, I was gonna say, before we get to that, this next question, I wanted to ask, because you said you actually earlier you said that the sea was a little choppy today, and maybe we talked about that before we actually started the session. And then if things are moving around, is this something that you were trained for when you come aboard the vessel? Because I would imagine in your classes you don't really get that kind of hands-on training.
It's pretty much, once you get here, they show you how to do it. They do ask who's experienced, and we have some coring technicians from USGS that are here with us. And of course, the ship has their own deck crew that are experienced. But every vessel's different how much they want the science team to participate. It's been really fun here because they really want us to participate. They don't have enough people, but they are interested. So they train you right then and there. You watch, you try. You get backed up the first couple times you try it. Someone experienced is nearby. But they just put a rope in your hand and say, hold on tight and good luck.
We have 3 people on board, and it's their first research cruise, and all 3 have been out on the back deck, and they've got to try and learn different things. And we usually start them off on a calmer day. and let the more experienced people do the rough days, but everybody has taken a turn on the back deck helping haul in that giant box core. Do we maybe want to play? We have a video of the box core. There's a couple more questions, but we can show you what it looks like when we're putting it overboard.
Perfect, Caitlin. Thank you. Yes. And thank you for the questions. Keep those coming. We're going to get to them.
Someone said, how long does it take to process a box core? I think we've been finishing after maybe 3 hours. We have a lot of different objectives and different scientists, and some people finish quickly, some people it takes a bit longer, but I'd say everyone's done in about 3 hours.
So here you can see us deploying the box core. See how massive it is.
There's
There's
Caitlin.
So when it's coming up, we hook it with these clips that are attached to lines, and then we can pull those lines tight, and that keeps it from swinging around, so we can kind of safely lower it on deck. We also have this A-frame that's moving in and out. So we have a lot of people that are trained on driving the A-frame, which is really fun. It's just this giant piece of equipment that you get to operate with a little joystick. And then you could see in that last clip, the box is up and all of the scientists are kind of rushed around it. So we all are excited to see what's in there and to start picking where we want to take our samples. We stick these long hollow tubes into the box, and then we take out individual cores. So some cores we measure oxygen, some cores we look at organic carbon, some cores we sieve and look for tiny worms. So all these different objectives get different cores from the same box. And then we have a ton of data, geological, geochemistry data, and also biological data coming from the same 1 meter square of seafloor.
You might have also seen that in the last section, they're fast little videos, but you might have seen us all stand around and hold hands around the box core. This has been a really wonderful experience for us. We have a cultural practitioner on board, Ku'upua Chang, who is from Manoa. And she has been helping us to take a moment to give thanks for accessing this part of the deep sea, a place that's very sacred to a lot of Hawaiians and Polynesians, and just to really remember and stop, because it's really, as a scientist, easy to just rush in and be so excited to get your hands in there and grab your sample. But we take a moment to all stand together and just kind of be grateful for the mud. And that's been a really wonderful practice to realize how lucky it is to get to access these really remote places that have a really big significance to a lot of different people.
And Ku'upua has a daily blog that we shared in this chat that she wanted to share with all of you. And I think it's going to be in an email after this meeting.
That's
so
right.
if you're interested, you can follow along with her daily blog.
And also maybe just before this portion of the video, I thought I saw some round samples in a container just very quickly. Is that what the ocean core samples look like?
There's like a pour water
Yeah.
video.
Yeah.
We have another video that we can see more of the processing. Here we go.
Yeah, so we subsample, I guess, the box core into what Caitlin was saying, these tubes. So you can see here, we're pulling these tubes that are filled with sediment out of the box core and capping both sides so the sediment doesn't fall out. So there, Kira is pushing a core in. So this is the cold van that you saw before. So after we take the cores, we quickly transfer them to this cold van so they stay cold. So any like microbes that are living there aren't dying, or the geochemistry isn't changing because of the hot temperatures. So here they're subsampling for fauna, I believe.
Oxygen's one thing that can change with temperature. So I'm using a special electrode to measure oxygen. And as soon as we take the core, we bring it up to that cold van and we do that work up in the freezing cold to keep those oxygen concentrations realistic. or accurate. It's a lot of fun playing in the mud. This is from yesterday. We're often covered in mud.
So we're sampling both for, a lot of groups are sampling both for sediment, but then also pore water, which is water within like the sediment grains. So here we're using what are called rhizons, which are little filters we're sticking into the core, and we're pulling the water out of the core. It's kind of like if you're like sucking out water through a straw. So here Maru has all these oh. See.
And it'll be really interesting in the future once we start generating all of this data, getting back together. We can kind of look at the chemistry of that pore water and the chemistry of the sediment. And then we can look at what microorganisms are living there and what animals are living there. And we can see how those two things are connected.
One of the in that kind of the last frame, I think there was one person who put like a sample container up to her eye. Is she actually looking at, is that like a microscope that she can see what's in the sample?
Yeah, so that's called a refractometer, and it's a quick, easy way to measure salinity. So what she's doing is taking a tiny drop of the pore water, putting it on a little disc, and then holding it up to the light, and then you have a little bit of scale that tells you salinity units. So sometimes we're collecting data, measuring and writing things down. Other times we're collecting samples that get frozen or put in the refrigerator, shipped back home to a lab and processed at home. I don't know if you guys can hear that winding sound, but I think we're on station now to deploy the box core. I feel like we've stopped moving a little bit and things are turning on.
Yeah.
We have something called dynamic positioning, which the captain and the bridge use to keep the boat in place so we can stay on station. So we've been on board long enough that I'm recognizing the screeches and the noises, and yeah, I think they're going to deploy soon.
Yeah, that's a funny thing. Whenever you come back to shore, the regular world seems so quiet compared to a ship. There's loud sounds everywhere here.
Well, I had to say the Zoom is doing its job in muting out all those ambient sounds. And in fact, I wish we could hear it because that's a part of the life on board the ship. Which is amazing. Yes. And Peter has said, oh, can you all give us a quick introduction to Peter? He's not there in frame with you, but he's one of your colleagues.
Yeah. Peter, do you want to unmute and say hi? Maybe not. He might be in a lab space.
I don't know if you guys can hear me.
Yes.
Hi, everyone. I'm just watching in the background. I helped to sort of put the visuals together, but I'm very excited to be here. And yeah, you guys are doing great.
Yes, they are doing wonderfully. And thank you for those video clips. It's really helping to immerse us in the experience and what your everyday life is like aboard the vessel, Peter. Thank you so much.
We're really lucky to have Peter on board to help us document the science that we're doing and to communicate the science that we're doing. And we often sail with a science communicator or an artist or some kind of visual storyteller to help us translate the science and the technical into things that everybody can understand and appreciate. So there are a lot of different ways to go to see if you're interested. We have this kind of diverse group of scientific backgrounds, but then we have Peter, our videographer. We have engineers who are keeping the ship running. We have our cooks who are making us meals every day. We have our officers who are navigating and driving the vessel from the bridge. So there's a lot of different pathways to getting to sail on a boat like this.
Absolutely. And that's definitely something I really want to make sure we highlight for our audience here and for those who will be watching the edited recording of this session, because there are many ways to contribute to the success of this kind of expedition. And I'm sure we'll talk a little bit more about that as we continue. For now, we have a few more questions in the chat, starting with Christian's questions there. How often does the box core fail to gather its sediment? Do you reattempt after a failed attempt?
That's a great question. And as the chief scientist, I'm the one tasked with making those decisions. It's really, you start to get a feel for, we get some clues as to whether or not the box core failed, because you have to decide, like, did the box core not happen right? Did something go wrong? Or is it a kind of sample we're not going to be able to collect with the box core? So a big box core like we have is designed for softer sediments. It's supposed to be able to land on its little landers and then trip down into the sediment and pick up a nice big box. But what we weren't necessarily expecting, but the deep sea is a big mystery and has a lot of different environments, is that some of the places where we've tried to land the box core and expected soft sediments, we found quite compacted, much harder sediments. So in the beginning, the box core would sort of try to go down, but couldn't get very much sample, or it's so hard that it's not getting any sample. So if it's a region we're really interested in, other factors like the mapping data or the oceanography data tell us we really, really want a sample here and we don't get a sample, there have been a couple times where I said, go back in, I really want to know about this region. I can't understand, geologically why we wouldn't get a sample, and we throw it back in. But sometimes as we get more experience and we start to characterize the region and see, well, in some places we really just have this compact sediment that the box core is the wrong tool for gathering that sediment, and it's not the kind that we're as interested in scientifically. I've started to feel more confident saying, okay, I'm just gonna get that data point. Like, no box core is its own data in a sense. We know that the seafloor type is not conducive to our sampling, so we make that observation and move on to the next site. Because like Caitlin was saying, with 5-hour wire times, where that's what we call it, when we're sending the box core down and waiting for it to come up, at some point you are sacrificing another data point for another. So anyways, that's probably too long of an answer, but that's a key part of our science that we're doing here, is like interpreting the samples, but sometimes no sample is also giving us quite a lot of information.
Oh, I think that was a perfectly detailed response. Thank you. That's exactly what we're looking for. And you all keep those questions coming. So from Arjun, this is a question from Finn. Has there been any malfunction or box core that's gotten stuck where you got to get out there and jump on top and try to pull it up? I added that extra part in from Finn.
Yeah. Yes, actually, there's been a couple exciting malfunctions. When we let the box core fall, you have to remember that not only is the box core at 5,500 meters, but you also have 5,500 meters of wire between the box and the ship. And in order to let it sink, and you also have the ship moving up and down. And so in order to let the box core get to the bottom and sink down into the sediment, you have to spool out some extra wire. And so that is probably one of the most difficult and dangerous parts of the operation, because we can't see. We don't have a live video feed of how that wire is moving around above the box core as the ocean is flowing and the ship is moving. And so one example of a failure is a wrap of wire got stuck around. There was an instrument called a USBL that tells us the exact location of the box core was mounted on with a steel mount onto the side of the box core frame. And a piece of wire got stuck in between the mount and the frame. And so we actually pulled the box core up from the seafloor, not from the safe top of the box core column where we're supposed to, but around that wrap. Luckily, the shear strength of that steel mount was very good. It came up just slightly bent out of place, and we didn't lose any instruments, but it did break a mount for our tilt sensor that tells us about how the box core is moving in the water. So that was sort of the box core didn't even trip. It was kind of a miracle that we got it back. So that's another example of changing things up at sea. Our coring technicians decided, okay, we're not going to mount it there anymore because that's too much of a hazard. They took it off, and now we've been mounting that USBL sensor onto the wire about 50 meters above the box core, so there's no worry about that happening again.
We can't see what's happening underwater during deployment. And so when we're recovering, we're standing at the back of the vessel, looking overboard, looking into the water. And that's when we learn. That's when we see the issue Kira was talking about during recovery. Okay, we have an issue. It looks a little bit different. And then we have to kind of pause and talk about how we're going to safely bring it back on board. What are the risks? And like Kira said, we're so lucky to have a lot of expertise and experience and people who know how to safely work with these different problems as they come up.
But out of 21 deployments, we've only had 2 major issues. So our batting average isn't too bad.
No, that it sounds like on every expedition there has to be things that are always going to happen, and that you just have to be ready and prepared to deal with any situation that comes up. And I mean, I imagine that that's just learning on your feet, and getting these kinds of experiences under your belt helps to build your confidence and your ability to successfully address any issues that come up, which
It
is
also takes a lot of teamwork too. So you kind of learn who you're working with. Everybody has unique and different skills. When you look over the edge and the box core is coming up in a way that doesn't look quite right, you have to, know who to turn to, let everybody kind of give their input and work together to bring it back on board.
brilliant. Teamwork and cooperation is the norm pretty much for all scientific research. No matter what you're doing, everyone's working together. You depend on each other for the success of the research. And that's definitely something we need to point out, because there's so much when we're students that we do on our own in labs. We may work with a partner, but when we're out in the real world professionally, cooperation is what makes a successful research team. So thank you for demonstrating that. And we see that with you all right here, which is amazing. Mackenzie has a question about eDNA. Is that used to help identify what's in the sediment?
Take that one.
Yeah, so we're taking eDNA samples from the water column, but also the sediment, right? So we have some ecologists on board that are like live identifying organisms as they come up, but then we're also collecting eDNA to hopefully pair with the live organisms that are being identified. And the eDNA will also help us identify organisms that we can't, actually see with our eyes.
Oh,
Yeah,
how, yeah,
sorry,
can you explain how that eDNA helps you identify sediment that, specimens that you don't see?
Yeah, so right now I'm shedding skin cells and hair, and my DNA is probably all over this bench. And animals in the ocean do the same thing. And so the CTD that we looked at earlier, where we collect water, we'll close those bottles. And then we have people on board that filter the water through this really fine little filter. And then they'll process that filter and sequence the DNA, piece it together, and try to identify, I know it's mind-blowing, try to identify different animals that were swimming through the water when we were taking that sample. A little bit of deep sea detective work.
Yeah, it's been a really, especially, I mean, oceanography is like, we're only out here for 30 days. We only get to send a thing down every 5 hours. So you would miss a lot of unless you happen to see it float by right at that second, which is how a lot of traditional species identification was done. eDNA lets us see who's not necessarily there at the exact moment that we're sampling. So we're doing that for both the water column and in the sediments. And the sediments we're really interested in because we haven't been getting as many live animals as we expected. Something we thought was that we'd get worms. There's a lot of different kinds of deep sea invertebrates and worms, and we have these specialists on board. But the regions where we are, there's not a lot of food available. And so we're finding that that's not supporting really robust benthic fauna communities. But maybe the eDNA can help tell us about who was there in the recent past, or animals that move through because there's not a lot of food. Maybe the animals are more mobile. Like, I'm just guessing here, but eDNA will help us to answer some of the questions that are remaining from what we're not seeing in the individual box scores we're getting ourselves.
eDNA seems to be something that's fairly new as a tool to use in marine science research. As students, Marissa and Caitlin, well, Caitlin, you're not anymore, but still, when you were students, and you still are, Marissa, were you trained on using eDNA for research?
I've never used eDNA. I study bacteria, which doesn't use eDNA. It does, I guess, just looking at regular DNA and metagenomics. So this cruise is really cool because I have never sampled eDNA or really learned about it. So it's something, really interesting to me that I was able to learn on this cruise.
And you mentioned the eDNA being kind of new. One of our scientists on board is collecting animal samples that she will preserve, and then they go into a collection that can be used for science. But part of that process is collecting a little tissue sample that she will then sequence. So we have the animal preserved, we have really nice photographs, a lot of information on the animal, and then we're also generating new DNA sequences, and that goes into the database that people are searching to try to identify these animals. So it is a relatively new technique, and we're still adding information to that database. So when someone finds a little piece of DNA, we can do a better job at identifying what it belongs to.
That's true. Like an eDNA sample is only as good as the library that you check it against, because you can't just know everything unless you have a species that you can fingerprint with. So we're kind of using that system, like Caitlin said, from both angles. Collecting eDNA to search, and also collecting animals that can be in the database that people search as they collect eDNA.
Yeah.
Wow, that's brilliant. That's incredible, incredible. Okay, I mean, let's get to our next questions here. Thank you all for these great questions. Okay, so we have here a question about opportunities for undergraduates to participate in the expedition. So maybe not necessarily just this expedition, but expeditions in general. Kira, are there opportunities for undergraduate students, community college students, to participate in some way?
Yeah, absolutely. Every time a cruise spools up, they're always looking for people that are interested in sailing. If you're an undergraduate and you have an advisor that is plugged in, I'm sort of, it's all sort of like case by case basis. What kind of science is happening? How many berths are available for additional students? But if you're someone who is really interested in trying out an oceanographic research expedition, try to connect with professors and teachers that you know are in that field and might be working adjacent to it. You can, there's also some programs through things like there are the Schmidt Ocean Institute and the Ocean Exploration Trust that runs the Nautilus has specific opportunities for undergraduates. Maybe Caitlin, you have more specifics.
Yeah, I was just gonna say I did my first 2 research cruises as an undergraduate, and I was working as an undergraduate research assistant. And I was lucky to have funding to do that. So the person that I was working for had funding to pay me as a research scientist and invited me to come out to sea. Not every professor has funding to pay you and hire you, but a lot are willing to take you on as volunteers, which I know is tough because you're all super busy. But if you have a little bit of time to volunteer and you're really interested in going to sea, I would encourage you to reach out to a professor and say, hey, we do a lot of bottle washing, and we were talking about the packing for this cruise. There's a lot of work that goes into prepping for expeditions, and a lot of that work is done by undergraduates. So if you can find someone who's willing to take you on as a volunteer, or even better, a paid technician, that could be one pathway. And as Kira mentioned, there are some programs like Nautilus Live and Schmidt Ocean Institute. Those opportunities are geared more towards interest in science communication. But we have a couple of undergraduate students that came on board kind of last minute because we had someone who couldn't come and we needed to fill a spot. Oh, sorry, they're graduate students. But
We
yeah,
have recent graduates, or
yeah.
Yeah, we have two people on board who have their bachelor's degree but are not in grad school, but are working in a science adjacent, in technician roles. So I would just say reach out to teachers and professors working in oceanography. and tell them that you're interested.
That's exactly the kind of information we want to get out there so that we can have students, those who want to be in these kinds of STEM careers, can really understand what pathways there are for them to get this kind of experience, and letting them know that you've experienced things, these things too. So it is possible, and you definitely have to keep trying to figure out a way to actually get there, to be where you are in one way or another. So students, if you have more questions about this, please put those in the chat. Let's continue with Vani. Vani's question is, what's been the most exciting discovery you've made so far? And not necessarily limited to this current expedition, but just through your career so far. Kira, and then Caitlin, Marissa, what's been the most exciting?
That's a really great question. I think because I'm fresh, because this is my baby, since I'm the chief scientist, I think my favorite box score we pulled up so far was actually last night. We pulled up a giant box full of something called silicious ooze. So there are organisms in the seawater that there are phytoplankton at the surface of the sea that they make their shells for their body out of oh, she's got a slide about it. They make their body out of silicate, so they're basically kind of have frustules made out of glass. But I myself, because I mostly study minerals, like actual rock formations, I'm learning a lot about deep sea sediments on this expedition. And so on the very right of Marissa's screen that she's just pulled up is an example. You can see how the core looks kind of white. It almost looks kind of foamy. And that was actually all the frustules of these small organisms, they run into each other and get stuck together like a miniature Velcro. And they were creating this sort of glass foam, basically. Like we were able to, get our cores out very easily. It was just a really neat experience to see that. And we really weren't necessarily, we'd been pulling up mud or nothing. And it was just this random depression in the seafloor full of this material. So we have a lot to learn. And Marissa has some specific science objectives that she's going to follow up with on mercury chemistry related to that. So it was just a really fun surprise that I wasn't expecting.
Oh, that I'd imagine that is sometimes not the case. You know what you're expecting to get in these things. So to have something that's unexpected, that brings a smile to your face and is so scientifically intriguing, it definitely sounds like it's a treat. Thank you, Kira. Caitlin, what about you?
My favorite thing so far on this expedition has been a carnivorous sponge. And so as Kira was mentioning, we haven't been finding a lot of animals on this expedition. The deep sea is a really tough place to live, and we're working in an area where there's just not a lot of food. But on one of the box score recoveries, we saw something that looked like a little stick. It was about this long. And so we brought out our animal person to take it into the lab, take a better look, and she was able to identify it as a carnivorous sponge. So a sponge that eats other animals. And I think that is so cool because we're not seeing a lot of food on the seafloor. We're not seeing a lot of animals, but this one mighty sponge is down there surviving and figuring out a way to get food. And she also mentioned that it hasn't been observed in, I think,
since the 1880s.
Since the 1880s, no one has found this particular species of sponge that we just happened to put our box score down on top. So that has been the most exciting for me.
Or maybe that was the first record.
The first, but I don't think a lot of them have been recorded in the ocean. Don't quote me on it, I guess.
Yeah.
We're not the animal team.
Yeah. But it was literally like one of the first drawings was from this region in the 1800s.
Incredible, a carnivorous sponge. So just to go along with what you were saying a bit further, you've mentioned multiple times that there's not a lot of food in this area, and that it's really challenging to live in the deep ocean. Do you expect a certain amount just in general of animal life in this area?
We really don't know. You know, no one's looked here before. So we're learning live in live time.
Just like us with you. So thank you for that. Marissa, so for you, the most exciting thing you've discovered so far?
Well, I also loved last night's box core and was super excited about that. But honestly, I get jazzed about every box core, just because there's not a lot of, so I study mercury cycling in the ocean, and there's not a lot of data on different forms of mercury. So there are 4 forms of mercury in ocean sediments. So really every core is like really exciting because that means it's like a new data point of mercury concentrations. And we're actually measuring 2 forms of mercury that haven't ever been measured in sediment before. So, and hopefully we'll be able to, it all works out, but.
Could you talk to us about that just a bit more before we get to the next question? You said you are doing mercury cycling. You're looking at that. What is mercury cycling?
I can actually show you some slides if that
Please.
be helpful. So maybe you've heard of like mercury in fish or mercury in thermometers. So there's actually mercury in the water column, and it's released into the environment through volcanic emissions, by coal plants, by mines. And once it gets into the water column, different microbes and minerals can transform mercury into different forms. So there's actually 4 forms of mercury. So I've listed them on the right here. And the reason why we care about mercury is because one of the forms, methyl mercury, can bioaccumulate in fish. So as a fish gets older or as you move up the food web, there are higher concentrations of methyl mercury. And then we eat fish and methyl mercury can be toxic to us. So it's really important to understand, mercury cycling in the ocean and in the sediments, because mercury actually in the water column absorbs to different particles and like dead organisms and falls to the seafloor. So there's a lot of mercury in the sediment. So one thing I'm really interested in is the cycling of mercury between these 4 different forms and kind of like untangling what's going on in these sediments.
So I know here living in Hawaii, it's I've been spoiled being able to get such good fresh fish all the time. But on the mainland, I certainly have family members who don't eat a lot of fish anymore because of the mercury in the fish. So this is the methyl mercury that you were saying that can build up in the fish over time.
Yep, and there's usually higher methyl mercury in freshwater fish, so that's why maybe in the lower 48 you hear more about that. But like in tuna, it's lower concentrations, and I mean it only becomes harmful if you're eating like a lot of fish every single day. Like I still eat fish, plenty of it.
Okay, and so since you're an expert, I'll feel okay eating
Yeah.
fish sometime. All right, good, good.
But I'm really excited to, have this data and pair it with like Caitlin and Kira's mineralogy data and some DNA looking at different microbes that are transforming this mercury.
Let's see here. Okay. Yes, thank you. Thank you. Sorry, I had a direct question sent to me. Yes, this was about the recording. Yes, the recording will be available to everyone on our YouTube channel. I promise that I will send it to all of you who are registered. Okay, so I know we have about 17 minutes left, so let's get to the rest of these questions. I wish we had more time with you, but thank you for fitting us in, because I know that things kind of change quickly, and it's just a pleasure to be here for you. So seconding, we have Christian seconding the undergraduate questions and says, I personally lack direction for research, but would like to be a part of something similar to this, but don't know where to start. And so I believe you've really already addressed that. And so if Christian, if there's, additional questions, I certainly can pass them along to Kira and Marissa and Caitlin for any additional help they could provide on that. Okay, another question from Vani, and thank you all for all the questions. These are great. Do the samples you collect give insight into the oceanographic history of this particular area? And I assume yes, because you said this particular space that you're in hasn't been studied before.
Yeah. And specifically sediments in the deep sea are a wonderful recorder of ocean chemistry. So as phytoplankton are living in the surface of the sea and then dying and sinking and being eaten, and then those animals are dying and sinking, and you have all these processes happening from the surface to the seafloor, and then you're basically building up layers of that in the sediment. So the sediment in this bottom of the ocean is made up of all the organisms and sediments that have been falling through the water column for centuries or for, millions of years. So by collecting cores of sediment and looking at that stratigraphy, which is basically the layering of the sediments, that is exactly the question that we can look at. We have some ways to think about age dating, like how old are the sediments, but even just by studying chemistry and mineralogy, we can see how conditions have changed over time based on what different kinds of sediments are being delivered to the seafloor and captured there. So it's a really good opportunity to study past ocean conditions, climates, chemistry, productivity, which is, what animals and plankton are living at the surface and then falling down to the seafloor. So that will be a really big deliverable from this expedition specifically.
Like a time capsule.
Yeah.
Yes. When do you actually open those core samples? Because I assume you do some on the ship, but afterwards, when you do your data analysis, I'd imagine that you would share out the different samples to the members of the team so that they could do their own particular research. When does that happen, and how long do you have to do processing before you come back together to share results?
That's a really good question. I would say like after a cruise, it takes at least several months up to a year, sometimes years to really like get the time. I mean, that's the exciting thing about science is we're always trying to balance like preparing for the next expedition versus working up the old data. So like Caitlin, for example, this is her fourth time going to sea this year, third time? Third time just in a year. So people like Caitlin who are collecting data multiple times a year, she's prepping for the next cruise before she's even really gotten a chance to look at the old stuff. And so, but in terms of like the samples themselves and their timeline, I know it depends on the analysis. Like for studying mineralogy, the minerals in these kinds of cores are pretty stable. So as long as we keep them refrigerated, there's not a time component for how quickly we need to process them. Sometimes we're working in regions where we're working with sulfides, which are very sensitive to changes when they get oxygenated. And so when they're brought up to the sea surface and living in the oxygen-rich environment that we greatly benefit from as oxygen-breathing humans, that then you have chemical changes and mineralogical changes happening in your cores, and we have to process those very quickly so that we can capture the in situ or the in-place conditions as quickly as possible. So for this, but then there are some analyses that people will want to get going really quickly. You don't want your DNA to degrade, but we can freeze DNA samples. So it'll take quite a long time. I think most post-cruise meetings happen maybe 6 months to a year after the expedition to start saying, okay, what did you find in your DNA? What did you find in the chemistry? What did you find in the mercury? So science is a long process.
We kind of keep writing papers and learning new things from the data set too. So after 6 months to a year, we've all kind of analyzed our samples. We understand the samples that we took, but then we start talking to each other and sharing that and connecting different things. And then, maybe Marissa and I are going to write a paper on, I found something exciting, she found something exciting, and now we're going to work on this. And so you kind of start coming together, and oftentimes several years after a research expedition, you're still writing papers as you're connecting these different data sets.
Well, and a big part of USGS and really oceanography in general is that people recognize how difficult it is to get these expeditions. They're very expensive, very time consuming, very difficult work. So there's a lot of effort to preserve as much data as possible. So the USGS has a core repository. So 1/2 of our cores will get put into a repository that then is publicly accessible to anybody in the world. And most, whether it's an academic institution, most people try to archive any archivable material so that it's available to people. So like I've written papers from cores collected or rocks collected in the 1980s. So, so somebody later might go somewhere similar in the ocean and say, oh, I want to compare it to the data south of Hawaii. I'm going to go access this core. Or like we talked about with DNA samples for people who are working on carnivorous sponges, they'll be able years from now to access the sponge that we collect. So yeah, sharing the data because it's such hard to access places is a really important part of oceanographic research.
And that's a great segue into our next question from Vani. What classes do you suggest an aspiring oceanographer focus on? And may I start first with, because you mentioned Caitlin and Marissa working on papers together, is how important is scientific writing in the work that you do? Because I know as my background is astronomy, and so that was one of the first classes we had to take was scientific writing.
Yeah, what classes should you take? I have a confession. I don't actually have a degree in oceanography, even though I'm an oceanographer. I didn't even go to school by the ocean. So my bachelor's and my PhD are from Wright State University in Dayton, Ohio, and my degrees are in environmental sciences. So you don't actually have to study oceanography to become an oceanographer. Like I mentioned, our interdisciplinary team, we have geologist and biologist and chemist. And even there's a lot of technology and engineering pathways into oceanography too. So I would recommend taking the science classes that you're interested in. If it's chemistry, math, physics, biology, take those core science classes because those are really important. Try some oceanography classes if there's kind of a field class, and science communication is really important too. I had a great science communication class where we learned how to write papers and abstracts, how to make PowerPoint slides that aren't super boring, how to make posters for conferences, kind of all different media of communicating science in an academic way.
I have an undergraduate degree in biology and also went to school in Wisconsin. So I took more freshwater courses, but I think the field classes really helped me and really is what really interested me. So I took, biogeochemistry, water chemistry classes, which then led me to my PhD.
Yeah, I think I'll just second the icing on the cake, which is I think there's a lot of emphasis on like marine biology in the general when you're getting your general education, and really even then like big animals, fish, all the things you can see and grab with your hand up to whales. And those things are amazing, but the ocean makes up the majority of our planet and is made out of chemicals, it's made out of rocks, it's made out of minerals, it's made out of mud. So if you're really interested in oceanography, you'll still get to hang out with people who study big animals and even see them yourself. But there's a whole vast field of oceanography outside of charismatic megafauna. That's what we call it. The like cool things we love, like dolphins. If that's your passion, go for it. But like there are so many people studying oceanography that specialize in all other different kinds of science. Even the physics of the ocean, you could study how waves move, how currents move, things you can't even see, or take in the lab. So if you're interested in the work, broaden your horizons to the very complex system that makes up the oceans.
We often use robotic tools to study the deep sea too. So we're using the box core on this expedition But Kira mentioned this is my third cruise. So my last cruise, we used a submarine to study the deep sea. And the cruise before, we used something called a remotely operated vehicle, which is this really cool deep sea robot that can go all the way down to the bottom of the ocean. And it has robot arms to collect samples and video. And that's one of our most powerful tools in deep sea science. And the people that operate those tools are engineers and computer programmers. And so maybe math and biology is not your thing. There are those pathways as well, robotics and engineering.
It seems like there are a lot of ways to get into doing the work that you do, and that's really exciting to know. It's because there are fields where you have to have very, very specific knowledge, and the pathway to getting there and being successful can be very, very narrow. But you're sharing with us that that's quite different in this field, and that's really exciting. All right. So for Maya, she says, I'm not sure if I missed this, but there was an interest in seeing if the manganese nodules were present in this area of the ocean. Have you found any yet?
Great question, Maya. I think she is an insider. I'm just kidding. Yes, so manganese nodules that's in the name of our expedition. They are little mineral concretions that look like maybe about the size of a fist that in many places in the abyssal plains can exist. And they were looking for manganese nodules, and the ecosystems that are associated with these kinds of deep sea minerals was a huge driver for this expedition. The USGS makes models of where we think minerals might occur, and there was a small region in the southern portion of the US waters south of Hawaii, which is where we're working, that we thought we might find them. We have not found any, which initially was a big disappointment because I study these kinds of minerals. I was very excited to check them out. But we're probing in an area that's completely unexplored, and learning about the fact that there are no nodules here is actually a really important data set for us to improve our models, for us to understand the heterogeneity of abyssal plains. It's really easy in science to go where you know the thing is, that it's there, and to go and study it. And that's a really important pathway. But it's also a really unique opportunity to probe the outer boundaries of where we think things might exist and might occur, and to gather that data and feed it back in. So we haven't found any nodules. We have about 3 days left and about 7 box cores remaining. So maybe we'll get some at the end. I think everyone would be very excited because they're very cool rocks. But in the end, we haven't found them here, and that's okay. Sometimes a negative result is your most important result.
Absolutely, absolutely. Let's keep going here. We have 4 minutes left, and I want to get to all the rest of the questions. So Vonnie asks, what technologies or instruments are most helpful to your research? And I know, Caitlin, you were just saying the underwater kind of submarine is really a fantastic tool that you all have used. Is there one more kind of tool that is maybe indispensable to this kind of research?
I would say seafloor mapping. And that's also something we didn't talk about much before, which is, there were 2 big expeditions that happened in advance of this research cruise, to do bathymetric mapping, which is from the ship. So they have on the bottom of the ship, they mount a multi-beam system that sends waves down to the seafloor, and then based on the topography, the waves come back, and you get information about seafloor roughness. You can see underwater mountains, underwater hills. And I have spent so many hours staring at the maps that we collected in this region, trying to pick different geologic regions, compare them with places where people have found nodules and other things. And so that base layer of mapping, even from ship-based, is really, really important. We already feel like we're sort of going without a ton of information, but if we didn't have the mapping, and we might drop the box core on top of us underwater mountain and break it the first time, so it's important that we have that base layer of mapping. But otherwise, like, we need samples, we need imagery, so all the tools that help us get those things take us to the next level. But I would say, like, mapping preliminary seafloor mapping data is indispensable.
And, we see that in astronomy too, mapping the southern polar region of the moon before we send an expedition of humans there to look for, different forms of water. Those are some invaluable preparatory steps that are also extremely necessary if you want to have high success at achieving those goals. Wonderful, wonderful. And thank you, Marissa, for pointing out the Seven Seas Media for undergrad opportunities. So I hope you all would check those out. What makes the abyssal plain nodules significant to your research?
Well, I mean, a big portion of the people here are part of the USGS's Seabed Mineral Resources research team. And so we study all currently forming kinds of mineral occurrences in the deep ocean. We want to know about the processes that help these mineral forms. We want to know about their resource potential. We want to know about how they contribute to the study of past ocean climates. They're collecting metals from seawater. They tell us about where metals are stored in the deep ocean and the balance between seawater and the surface water and the deep ocean. So my whole job is studying nodules, studying ferromanganese crusts, seafloor massive sulfides, any kind of minerals that are forming at the seafloor in the modern ocean. So it's significant because that's my job, is to study those kinds of things.
Absolutely fair, yes. Okay, and then the most intriguing insights you have that your research has given you into Earth's history, and I think this is a great question to wrap things up. Again, a question from Vonnie.
That's a big one.
Yes, and you're already having insights right now that you've shared that there's just not a lot of food in this area. I know this area of the ocean where you are hasn't been studied for a long time. Seeing that, finding that carnivorous sponge, first one since the 1800s. I mean, there's so many things that you're learning already just from this one expedition. I can only imagine that your previous research is giving you lots of various insights that connect to the evolution of the Earth in general, but our water systems, which, as you said, covers over 70% of our planet.
Yeah, I mean, I think so. For ferromanganese minerals like the nodules or the crust, they grow about 1 millimeter per million years. And I think that there's a sense that the deep ocean is sort of like a big bathtub that's been the same for the whole history of the Earth. But when you pull up a ferromanganese crust or a nodule, you can see, even with your own naked eye, like completely different layering structures that tell you about different ocean conditions, times when there were really fast moving currents and the mineral could precipitate very finely, or when the ocean was very sluggish and you have these different textures completely. So I think for me, the way that studying deep sea minerals, it reminds me that the Earth is very dynamic, and that over the millions of years that we've had our modern oceans, things have changed chemically and physically many times over. And really, in this region specifically, not finding nodules means that we're experiencing a different kind of seafloor condition than we could have imagined just looking at a flat map from the top. So